Hi everyone. Today we're going to explore and talk about chlorophylls. I'll try to explain as best as I can. So to get started, I want to bring up what plays an essential part in chlorophylls. The sun. We all know, at least I hope you know, how powerful the sun really is. Although humans can't use solar energy to carry out metabolic processes, plants can. And here we have our example plant of the day. To put it simply, plants are able to capture light energy and use it to make sugars through a process called photosynthesis. So, how does this work? The leaves of plants are made up of cells, and inside those cells are chloroplasts. This all begins inside these chloroplasts, more specifically inside the thylakoid membrane, which I brought up here, which is, you can see the chloroplast, and I'm zooming into that part of the thylakoids, which kind of look like pancakes. And this is where you will find the specialized light-absorbing pigments called chlorophylls. The primary pigments, which we're going to focus on today, are chlorophylls A and B. Secondary pigments include carotenoids such as beta carotene. We're not going to focus too much about it, but just know that they're secondary pigments. So let's talk about the structure of the primary pigments. Here you can see that they have a porphyrin ring head. And what this means is that it is a circular group of atoms that is surrounding a magnesium ion. This is similar to the structure of hemoglobin, which you might be familiar with, except there is a magnesium and not an iron in the center. They have a long vital side chain, which is hydrophobic. And this is important because it's able to insert itself into the thylakoid membrane. And the difference between chlorophyll A and chlorophyll B is that chlorophyll A has a methyl group right here, and chlorophyll B has an aldehyde instead. So let me outline those parts here. Here would be the methyl group. Here is the methyl group, and here is the aldehyde for chlorophyll B. Also, another difference is that most plants have about twice as much chlorophyll A as they have chlorophyll B. And the ring system surrounding magnesium has alternating double and single bonds, which I hope you can see here. And this is important because it makes it a polyene. And this is particularly special because polyenes are able to strongly absorb light in the visible region of the spectrum, which is what we're going to talk about next. So here we have an electromagnetic spectrum, and it's important when trying to understand light energy. Light is a type of energy that travels in waves, and it's a form of electromagnetic radiation, just like microwaves and radio waves. It has its own particular wavelength, and what I want you to focus on is this part right here, which is the visible spectrum, and it contains the colors that we can all see. Well, most of us. Also, this is where photons come in, as they are particles of electromagnetic radiation, and they have a certain amount of energy depending on the wavelength that they have, depending on the wavelength that the energy has. So here, I made a little graph for you, and I wanted to show you the absorption of visible light by photopigments. In this absorption spectrum, you can see that chlorophyll B and chlorophyll A both absorb light from the blue and red region of the spectrum. So you can see here that 400 nanometers of the wavelength is like at the blue region of the visible light spectrum, and 700 nanometers along with its distance to 600 nanometers is like the red region of the visible light spectrum. So this means that they absorb this light and reflect all others. So they reflect green light. This is why plants look green to us. So, okay, now that we have the structure and some basic concepts out of the way, let's go a little bit deeper. So, like I mentioned before, thylakoids are these flat structures inside chloroplasts, which kind of look like pancakes. And if we zoom in, you can see that chlorophylls, which you can see are the green sort of oval within the membrane. And they create lines which ultimately make up photosystems. So, one of the most important things is that pigment molecules can absorb photons. So, I'm talking about chlorophylls. They can absorb photons, but only a special couple of chlorophylls can actually turn light energy into chemical energy. And this has to occur in a specialized section called the photochemical reaction center. And it's what I have labeled there. So the other chlorophyll molecules just absorb photons, and they're called antenna molecules, which I have outlined there as well. So they're called that because essentially what they do is absorb the light energy and they transfer it rapidly to other chlorophylls until they reach the photochemical reaction center. So it's sort of like this bumping movement that the antenna molecules do, or like as I see it, it's like passing a baton, or if the molecules are playing like hot potato. So ultimately, like I mentioned, the antenna molecules transmit the energy to a chlorophyll in the reaction center, and this is called an exciton transfer. So what we have here is a sequential process where an electron goes up in energy and it relaxes back down. But when it relaxes back down to a ground state, it transfers that energy that it's released because it went back to its ground state, and it transfers that energy to nearby pigments, and it keeps doing that and doing that until one of the antenna chlorophylls meets a pair of the chlorophyll molecules at the photochemical reaction center, which I have shown there. So once again, exciton transfer is when a quantum of energy is passed from an excited molecule to another molecule. So in this case would be the chlorophylls or the antenna molecules. So lastly, this photoexcitation results in a charge separation in the reaction center. And this begins the oxidation reduction chain that is going to be necessary to create an electrochemical potential, which will ultimately drive ATP synthesis. So that's the summary. And that's it. Today you learned about chlorophylls, their structure and their functions. And lastly, I just want to mention that all the drawings in this presentation are modified or redrawn from my Principles of Biochemistry textbook, which is cited here below. I hope you enjoyed. Thanks for listening.